So, good morning, everybody, and welcome to Bridging Research and Medicine Translational Advances in Cancer Immunotherapy. Here at the La Jolla Institute for Immunology, we're really delighted to have you here with us today. I'm Dr. Sonia Sharma. I'm an associate professor in the Center for Cancer Immunotherapy here at LJI, and it's a pleasure to have you with us today. Before we begin our introduction of today's featured speakers for you, I'd like to invite to the podium right now the 2026 Swing for a Cure co-chair, Helen O'Sur, to share a few words, please.
Thank you, Dr. Sharma. Okay, before we get started, I just want to introduce myself. As you all know, my name is Helen O'Sur, and also I'm a co-chair with Carol Tessaccini, who we all love and admire, for Swing for a Cure at the Heights Golf Course. And before I say anything else, I would like to welcome you all and thank you for attending this informative educational event. We would also like to extend our sincere gratitude to La Jolla Institute of Immunology for organizing this wonderful seminar, and also to the speakers for educating and sharing your knowledge of translational advances in cancer immunotherapy. And finally, Carol and I would like to especially thank and extend our special appreciation to Sarah Felt Morales for her effort in coordinating this event. Thank you. And before I sit down, Carol make sure that all of you have this flyer, which is our Swing for a Cure tournament in March. Okay, thank you.
Thank you so much. Thank you so much for your thoughtful remarks. And again, I just want to express that we're so grateful to have you all here today. You're a true testament to the legacy of your late husband, Dr. Charlie Osur, a respected fellow immunologist, a pillar of the San Diego immunology community, and actually one of our very own faculty here at LJI from 2012 to 2017. We are also really honored to welcome our friends today from the Heights Golf Club in Rancho Bernardo. Since 2018, your community has raised over $225,000 to support research into precision immunotherapy for cancer, right here at LJI. Funds that have directly advanced the work in my own lab since 2024 through your Swing for a Cure and Bingo for a Cure fundraisers. Thank you. The Bingo for a Cure fundraiser your club organized this past March supported my lab's efforts to improve both the effectiveness but also, importantly, the safety of immune checkpoint blockade cancer immunotherapy for patients who have solid tumor. These therapies that we're studying have the potential to transform cancer treatment, making it widely accessible and safer for all patients who might otherwise be susceptible to developing autoimmune disease during their cancer immunotherapy treatment, which essentially would end their therapy while they would need to be treated for that autoimmune disease. My team has been studying blood samples from roughly 150 cancer immunotherapy patients who are receiving, for example, Keytruda and Yervoy. And what we discovered by profiling the blood of these patients was we found that those patients who experienced severe inflammatory events during their Keytruda or Yervoy therapy, colitis, lung inflammation, type 1 diabetes, hepatitis, they all had significantly lower levels of an anti-inflammatory molecule called LPC. LPC is a metabolite. It's a fat actually, something that our bodies make from the fats that we ingest in our food every day. And we found that patients who had low levels of LPC developed inflammation through specifically the activity of a specific type of inflammatory cell called a neutrophil. LPC actually controls neutrophils and is involved in bringing down that inflammation. And when it's not there, these patients can succumb to these inflammatory events, particularly during their cancer immunotherapy. Building on this insight, we've actually begun to test LPC supplementation, and we've actually found promising results. Not only does LPC reduce inflammation, but rather unexpectedly, actually, we're actually finding that in preclinical models that LPC is actually increasing the effectiveness of the cancer immunotherapy in shrinking the size of those tumors. And again, it goes back to those neutrophils, which are good cells when you have a bacterial or a viral infection, but if they stick around too long, they can have an inflammatory and also a pro-tumoral role. So the work of LPC in controlling these cells is actually boosting the effectiveness of the cancer immunotherapy. I just want to say, switching from the basic research I'm sharing with you, but to really say that this research is truly fueled by memory. It's fueled by loss, by all those that we've all loved and lost to this disease, cancer, who yet still live on in our memories and our hearts, and I think inspire us because we're all connected by that to kind of work together in ways to keep pushing the research forward. Because cancer research, as you know, you'll hear by the folks who are speaking today, um, cancer research, I'm always proud to say this, it works at an incredibly quick rate. Things are moving and they're moving fast and they're having results. But we want to continue moving that quickly because at the end of the day, that's what is going to help the patients. It can be transformative. And it really comes from this deep, unrelenting resolve for us to actually conquer this disease. And in saying that, I just want to stress how the progress is a collective milestone. And I just want to sincerely thank the organizing committee for their dedication for hosting these fundraisers year after year, and to the members, to their families, and to the friends whose generosity and support, whose time make this work possible. And for the upcoming 2026 Swing for a Cure fundraiser, the organizing committee, led by Helen and Carol, had the idea to expand the fundraiser's impact through education, and that brings us all here today, speaking with all of you. For nearly 8 years, the members of the Heights have really championed LJI through their philanthropic support. And now, by bringing this educational seminar to life in this way, you're actually helping to broaden awareness in the community. These are the latest advances in cancer immunotherapy happening right here at LJI, an institute that is exclusively dedicated to studying the intricacies of the immune system. So on behalf of LJI and my lab members, some of whom are here today, I just want to thank Helen, Carol, and the entire Swing for a Cure committee for their vision, their dedication, and the continued partnership. Your presence here today really reflects a shared belief in what can be achieved when scientific inquiry is nurtured, when it's sustained, and when it's guided by collaboration. Progress in immune-driven medicine doesn't happen in isolation. It happens through partnership, through shared purpose, and the unwavering commitment to translating discoveries that we make here into better care for patients ultimately. This afternoon, we have two presenters lined up for you whom I will introduce shortly. And we ask that you kindly hold your questions until both presentations have concluded. And at that time, a microphone will be passed around for anyone who would like to ask their question, share their comment, share their experience, or just say hello. We're looking forward to a thoughtful and engaging discussion with you all, and we appreciate your participation. And so now it's my privilege to recognize two individuals who are close to me scientifically and as friends who are dedicating their time and their careers to advancing cancer research and therapeutics. We'll begin a presentation from Alicia Gibbons, a doctoral research in the Biomedical Sciences program at UC San Diego, who specializes in cancer and tumor immunology under my mentorship. Alicia graduated from UCLA in 2022 with a bachelor in science and microbiology and immunology, as well as molecular genetics. Alicia's promise as a future leader in the field was recognized early through receiving a very competitive BioLegend graduate fellowship. This is awarded to very competitive trainees. And her research focuses on enhancing the effectiveness of cancer immunotherapies while reducing those dangerous side effects that we talked about. She has also contributed to advancing our understanding of sex-based differences in the immune system, co-authoring a landmark science review that we recently published alongside myself and our president at LJI, Dr. Erica Ullman-Sapphire. That brought new attention to how biological sex actually shapes immune responses across tissues, including in tumors. Just over 2 weeks ago, Alicia won a Women's Health Access Matters WHAM Edge Award, which will fund her groundbreaking research to determine whether LPC, that anti-inflammatory molecule that we talked about, is changed in patients who are experiencing triple-negative breast cancer after they receive immunotherapy. Through this research, she aims to uncover whether LPC could perhaps ultimately serve as a biomarker as well as a modulator therapeutic that could predict who will experience severe side effects and then also treat those side effects. It could potentially be developed into a clinical test to help oncologists identify which patients are most likely to benefit from treatment while minimizing side effects and boosting anti-tumor response. Following Alicia's presentation, we'll hear from Dr. Sandip Patel, a fellow of the American Society of Clinical Oncology, or FASCO, an honor recognizing his extraordinary service and dedication to advancing patient care for cancer. Although we don't currently have a clinical oncologist here on staff at LJI, I am fortunate to work and collaborate with Dr. Patel, whose partnership has bridged our research into clinical application and clinical scope. Dr. Patel is a highly respected, board-certified medical oncologist who specializes in cancer immunotherapy and the development of early-phase clinical trials across all cancer types. His work focuses on truly developing personalized treatments for cancer care that activate the patient's own immune system to attack and target a tumor in a precise manner. At UC San Diego's Health Precision Immunotherapy Clinic, Dr. Patel leads efforts to bring the most promising investigational immunotherapies directly to patients. As I said, it moves quickly. As a professor in the Department of Medicine, he also mentors medical students, residents, fellows, while his research really focuses on identifying biomarkers that would guide the most effective, individualized treatment strategies for patients. He completed his fellowship in medical oncology and hematology at Duke University School of Medicine, his residency in internal medicine, at the UCLA Medical Center. So you're actually right. Thank you. And earned his medical degree from Baylor College of Medicine, where he also conducted research at MD Anderson Cancer Center. So we're honored to have both Alicia and Dr. Patel with us here today to share their work and their insights, demonstrating how collaborations between basic researchers and medical professionals can accelerate progress and bring hope and change to patients everywhere. So before we begin, I'd like to take a moment to introduce La Jolla Institute for Immunology, as this may be your first time visiting. So to begin there, LJI is an independent, nonprofit research institute that is dedicated solely to the understanding of the intricacies of the immune system. The immune system, it's really important to say this, it really drives every major disease, from cancer to infections to chronic inflammatory disorders, and the differences in how men and women experience illness. We here at LJI believe that actually every disease is an immune disease because every organ system is impacted by the immune cells and the immune system. To tackle this extreme breadth, LJI is organized into 4 distinct research centers, each one designed to focus expertise and foster collaborations. Our more than 200 scientists are working to harness the power of the immune system to develop better vaccines, new therapies for a range of immune diseases, and groundbreaking treatments for conditions that currently have no cure. LJI is purpose-built to surround these scientists with expert administrative support, allowing the investigators to dedicate their energy and their talent entirely to making new discoveries. This is a deliberate structure and is one of the key ways that LJI stands apart from its peers. The discoveries made here, they don't just stay in the lab. They move into clinical applications, helping to reshape how diseases are treated and, in many cases, prevented. We're honored to be the quiet force behind the innovations and tools that impact how you and your loved ones are diagnosed, treated, and cared for during life's most difficult health moments. We're actively advancing new diagnostics and therapies every single day, creating the innovations patients depend on today and also in the future. And with that, thank you for your attention, and it's my absolute pleasure to introduce Alicia Gibbons to the stage.
Can you hear me? Yes. Thank you, Sonia, for that kind introduction. So today I'll be talking about the research project that our lab is focusing on, wherein we're aiming to build better cancer immunotherapy. We have a laser pointer? So, another one. This one. Yeah.
You hold it at the top.
Oops.
Sorry.
Wrong button.
Wrong button.
Oh, okay, just have to hold it down.
Yeah,
okay,
awesome.
Great. So first I'll introduce myself. Thank you for the kind introduction. My name is Alicia. I'm a 4th year graduate student in the Biomedical Sciences PhD program at UCSD, and I joined the Sharma lab at the beginning of this year. So I originally grew up on the East Coast, and I moved to California to do my undergraduate at UCLA. As you heard, my major had a very long name, but that was just one major. Yeah, I loved SoCal so much I decided to stay here for my PhD. So I'm the first in my family to go to grad school and the first to pursue a career in research. Throughout my PhD, I've had a special opportunity to be a part of 2 research labs, and both have been focused on making cancer immunotherapy better. In the Sharma lab, we're studying how the immune system, or how we can make immunotherapy more tolerable specifically and safer for patients, to overall make cancer treatment better. And in the Sharma lab, we're also interested in studying the sex-specific differences in immunology. As Sonia mentioned, we recently published a review in Science discussing sex-specific differences in tissue immunity. And this is a picture of myself last Halloween. I dressed up as one of our chemical bottles. So where do we start? Let's talk about the pillars of cancer care. So cancer care is supported by 5 key pillars: surgery, radiotherapy, chemotherapy, molecular targeted therapy, and immunotherapy. Physicians use strategies from all 5 pillars in treating patients, And in the recent decades, the development of sequencing technologies and molecular profiling technologies have really given researchers the granularity to study the mechanisms by which cells signal and how they can be programmed or reprogrammed. This granularity has led to the development of more targeted cancer treatment strategies, including molecular targeted therapy and immunotherapy. Our research in the Sharma lab focuses on immunotherapy, wherein immune cells in the body are reprogrammed or engineered to fight tumor cells. Immunotherapy has transformed cancer treatment, and approaches including immune checkpoint blockade therapy are even now the standard of care for multiple cancer types. And in 2018, Dr. James P. Allison and Dr. Tezuko Honjo actually received the Nobel Prize in Physiology or Medicine for the development, their work in developing immune checkpoint blockade therapy. So what is immune checkpoint blockade therapy? Normally in the body, after immune cells become activated due to a threat such as a bacterial or viral infection, other cells will tell them to quiet down and to reduce their activation. This is important. Turning off immune cells' activation keeps them from mistakenly attacking other cells in the body and from causing chronic inflammation. But in the context of cancer, this can be detrimental. Killer T cells are one of the few types of cells that can directly target and kill cancer cells. But both cancer cells, as well as other immune cells, can express these inhibitory receptors that can turn T cells off. So you can imagine that this interaction, inhibitory interaction from the tumor cell to the T cell, or from other immune cells to the T cell, allows the tumor to escape death mediated by the T cell and can cause cancer to progress. In immune checkpoint blockade therapy, we attempt to block this inhibitory interaction. We use specialized antibodies that fit in between these two proteins, blocking the interaction, keeping T cells activated, and leading to tumor cell killing. So there are two main types of immune checkpoint blockade therapy. The first is anti-PD-L1 or anti-PD-1 therapy, wherein we block this interaction, this inhibitory interaction between the tumor cell or the killer T cell, or anti-CTLA4 therapy, where we move up the chain a little bit and we block inhibitory signaling between other immune cells and T cells, leaving the T cell activated and able to kill tumor cells in the body. Unfortunately, as Sonia mentioned, one drawback of immune checkpoint blockade therapy are these immune-related adverse events. Immune-related adverse events occur as a result of heightened immune activation because we blocked inhibitory signaling, and they present as tissue inflammation in a variety of organs through a variety of presentations. So patients may experience inflammation in a variety of tissues, and unfortunately, immune checkpoint blockade IRAEs are pretty common, with up to 30% of patients experiencing these severe grade 3 or 4 IRAEs, frequently leading to treatment discontinuation. Current standard of care treatment for these IRAEs include corticosteroids, which are limited in that they're broadly immunosuppressive and may affect treatment response. These IRAEs are not well understood. We don't really understand why they happen or how to treat them. So our lab decided to dive in on this question and investigate the central idea. What are the mediators of immune checkpoint blockade-induced IRAEs? So we focused on a specific subset of molecules in the body, termed the metabolome, which comprises an individual's pool of metabolites. Metabolites are small molecule derivatives of cellular processes and can also come from dietary intake. They have a variety of functions in the body, many of which remain poorly understood. As Sonia mentioned, we decided to leverage our proximity to the Morse Cancer Center and look at metabolites in real patients who were getting immune checkpoint blockade therapy for solid tumors. We collected blood plasma from patients, as well as information about their response to treatment and the incidence and prevalence of IRAEs. And then we performed metabolomics to investigate how the levels of thousands of specific metabolites changed over time in the blood of these patients. We've since been working on the functional characterization of these metabolites. So as Sonia mentioned, we pulled out two species of this LPC lipid group, LPC 18-2 and LPC 16-0. So these lipids, or LPCs, we found them to be inversely associated with the development of severe IRAEs, which could suggest that these metabolites are protective against IRAE development, as their loss is associated with IRAE presence. LPCs comprise a class of bioactive mediators involved in immunoregulation and cell signaling. They're complex molecules with both pro-inflammatory and anti-inflammatory roles and can directly modulate immune cells. As Sonia mentioned, we are hypothesizing that these LPC species can control IRAEs through their actions on neutrophils. Neutrophils are the most abundant blood cells, and they have complex roles in the body. Recent research has suggested, during times of inflammation, neutrophils can traffic to tissue, recruit to other immune cells, leading to a snowballing effect that amplifies tissue inflammation. Some recent research has suggested that tissue neutrophilia can be important in the development of IRAEs. So it was exciting to us when we saw that LPC 18-2 species, which was inversely associated with IRAE incidence, was also inversely associated with patient neutrophilia. So patients who had IRAEs tended to have more neutrophils, but they had less LPC, and that LPC was also inversely associated with their amount of neutrophils. So we decided to functionally test in mice using a special mouse model if LPC 18-2 could be protective against IRAEs through the control of neutrophils. And what we did here was induce IRAEs in mice using real immune checkpoint blockade antibodies. And we saw that the administration of the immune checkpoint blockade antibodies caused colon damage, indicative of a type of tissue IRAE, and also increased the amount of neutrophils both in the blood and in the large intestine. Excitingly, we found that when we co-administered the immune checkpoint blockade antibodies with LPC, that colon damage was alleviated and there was no more blood neutrophilia or tissue neutrophilia. So we found in these mice really that LPC 18-2 could prevent this IRAE effect, and it seemed like there was a relationship between that and its control of pathogenic neutrophils. Overall, we believe these data demonstrate that LPC has the potential to control IRAEs in both patients and mice through the control of these pathogenic neutrophils. We hope that this work has the potential to improve cancer immunotherapy for many thousands of patients who are undergoing immune checkpoint blockade therapies. Where do we go next? So I'm excited to share, as Sonia also mentioned, that I was recently awarded the WHAM Edge Award. WHAM, or Women's Health Access Matters, is an organization aiming to support research focused on sex-based health conditions. And we proposed an exciting project where we will aim to extend our LPC story to another type of cancer, triple-negative breast cancer, which is an aggressive and difficult to treat type of cancer that predominantly affects women. Over the next year, I will profile TNBC samples from Moores Cancer Center, and we'll also utilize a mouse model to investigate if LPC is correlated with treatment response or IRAE incidence in TNBC. I'm very excited for this project. Unfortunately, breast cancer runs in my family, and both of my grandmothers have passed away from the disease. So it's really a privilege to be able to work on something that's so personally meaningful and that has the potential to, help so many people. With that, I'd like to thank my team, the Sharma Lab, Dr. Sonia Sharma, as well as Dr. Musa Vatan-Makanian, talented postdoc in our lab. I would, of course, like to thank the Heights. We really appreciate your continued support of our research, especially during these uncertain times. And we're very grateful for your support through these projects. I'd like to also thank WHAM and BioLegend for personally funding me, as well as our other collaborators and funding sources. Thank you.
Thank you so much, Alicia, for sharing the work that you're doing and for helping us better understand the potential of the anti-inflammatory molecule LPC. Your insights in how it could help manage IRAEs and enhance patient quality of life during immune checkpoint blockade therapy are really fascinating, and it's really at the cutting edge of where cancer immunotherapy research is and should be. So building on that momentum, I'm really thrilled to welcome Dr. Sandip Patel, professor at the University of California, San Diego, medical director of the Clinical Research Informatics, leader of experimental therapeutics, co-leader of the Solid Tumor Therapeutics Program, and deputy director of the Sanford Stem Cell Clinical Center. Dr. Patel will share his insights into oncology, clinical trials, highlighting their goals and challenges that they face, and emerging next generation therapies. Thank you, Dr. Patel.
No, thank you so much. And I know you guys heard Wham and you thought George Michael was going to be next. Apologies, it's just me. I think that's a reference I only get. You probably don't know who George Michael is. So I'm going to talk about advances in oncology clinical trials from bench to bedside to the community and back. And I want to talk about clinical trials more broadly, and then lean into more maybe a verbal discussion around immune-related adverse events and some of the things we're trying to do to protect patients. We're probably in our most promising era of treatments. Not that we've been able to cure everybody or help everybody as much as we would like, but the idea that we have cancers that has 0% 5-year survival that now have 15 or 20%. And we want to get to 99, 100% as much as possible, but we still have to make progress. And anytime I'm having a discussion with a patient in clinic, everything's about the risk-benefit ratio, right? And with these immunotherapies, because we're unlocking a patient's own immune system, we have the potential for long-term benefit for their patients. But the immune system's a double-edged sword, and you can get autoimmune side effects, these immune-related adverse events that you heard about so nicely. So I wanted to give a bit about the clinical picture, and also how do we learn from clinical trials? Because the only way we're going to improve on our ability to treat patients with immunotherapies is to develop new immunotherapies. And these immunotherapies have different types of side effects. You heard about triple-negative breast cancer and immune-related adverse events. Some of the other clinical trials are doing have different types of side effects. So I'm going to discuss what we're trying to do to kind of systematically approach all these, really with the idea of really taking nice discoveries from the bench, like you heard about, taking them to the bedside for our patients, making sure they're available in the broader community. They don't always have to come to the university to get that kind of treatment, and how we can learn from each patient so that we can treat the next patient better. And so this is just a tour of our county, and I think San Diego and Imperial, even though we're neighbors, we're actually very different counties. And the reason this is important, I don't have Riverside up here, is we get patients from all 3 counties. We are at UCSD Morris Cancer Center, and La Jolla Institute is a key member of our cancer center, a matrix cancer center. Our job is to take care of everyone in the community in terms of research, clinical trials, and just our demography. And you can see for different ethnic populations, you see different proportions in the county. And even San Diego County, it's a very large county. North County is very different from East County, is very different from South County. I think the demographics I don't show here is that San Diego County is generally older than Imperial County. So we actually get a different distribution of cancers. For example, and this gets to even the molecular level. Let's say you're studying breast cancer. You're going to see more estrogen receptor positive breast cancer in San Diego County than in Imperial County, because it's a younger population. You're going to see more triple negative breast cancer or HER2-driven breast cancer. So this really helps us understand our population. So this isn't just a demographic exercise. This is us making sure we can take care of everyone going all the way to the Arizona border. And increasingly, we have patients flying in domestically and internationally for some of the more bespoke clinical trials that we do as well. And so really our fundamental goal with any clinical trial, especially at an institution like ours, which is a consortium institution that really tries to take discoveries all the way from the bench to the bedside, is to make these fundamental discoveries like you heard about, use basic science to improve human life by using these rapidly in clinical trials. And then once we know this works, making sure these are disseminated throughout the communities. Our campus in East County, near Alvarado, folks may have read about in the newspaper, Palomar, El Centro Regional Medical Center, really to make sure that we're able to touch lives in a positive way for folks all throughout the region. Education is a key aspect, right? The first talk was given, you could have been a faculty member, but in fact, it's a PhD student, right? And so really making sure we get the next generation of folks to really drive our discoveries, because fundamentally, I'm gonna be a patient one day as well, right? And so how do we make sure the next generation is there to do research, take care of folks? And how do we collaborate? We're really in a very wonderful ecosystem of industry collaborators, academic institutions, community partners, including what you guys do. How do we make sure that we bring these treatments as rapidly as possible to folks who need them for a disease that is as serious and as unfortunately rapid as we're fighting with cancer? And so one of the key parts of any exercise, and I always, when I'm talking with patients, it's always the balance of feeling like you're the test pilot, right, and not the guinea pig, right? That's the most important feeling in the world. For you, you are fighting cancer. This is your battle. It's the first thing you think about when you wake up. It's the last thing you think about when you go to bed. So what can we do to help folks, right? What can we do to help folks with their families? Now, one point about clinical trials is a well-designed clinical trial makes you feel like you're in control and you're the test pilot on that clinical trial. And so that's the way we try to design these studies to really help folks and make sure either it's a single arm study, which means whatever you do, you're getting the drug, or if it's a randomized controlled trial, you may have heard about these RCTs, that both arms are reasonable, meaning, it's Diet Coke versus Coke Zero, right? In terms of what you're picking, though I know folks may have some strong preferences there, though here it's just coffee, right? And so the question is, how do you design a trial? And it turns out the biggest barrier to accrual, you don't have to read in the small boxes. It's really about awareness, opportunity, and in giving the patient the power to accept or refuse. And this, to me, is the most important aspect. So everyone should be offered a clinical trial. Everyone should be offered all of their options, standard of care, as well as, for folks who may be more thinking about no chemotherapy, right? Thinking about comfort care, hospice, things like that, for the right patient. Thinking about the opportunity that a clinical trial develops, and then the opportunity to make the option, right? Between options A, B, C, D, if options A and B are standard of care and C and D are trials, well, what information can we provide to help make the informed decision? And what I always like to tell my patients, because everyone always feels like if I pick A, then I can't do C, is most of our decisions in oncology are the order in which we're going to do it, right? So it's A, then B, or B, then A. It's not necessarily A versus B. But the making sure we understand everything we're doing to try to help folks. And so historically, these have been very centralized approaches for clinical trials. You'd come to La Jolla, right? You'd get all your study assessments, you'd meet with the doctor, and you'd come in every couple weeks for your treatment. What we're trying to do is decentralize clinical trials, and this is called care at home. We're working on this. We're not quite there yet, but I want to talk about some of the things we're working on because it very much relates to how we handle immune-related adverse events. Because when we used to talk about side effects, we used to talk about chemotherapy. And the majority of the very severe chemotherapy side effects, and I'm not saying that the fatigue and the nausea at home is not severe, but the life-threatening side effects happen in the infusion center. And so that's the way the healthcare system is wired, because for decades, we were treating patients predominantly chemotherapy. But as we have newer treatments, and in fact, in my phase 1 career, I've never developed a chemotherapy drug. Where we're using the chemotherapy drugs we have and we're combining them or replacing them with these novel immunotherapies or targeted therapies. But we are trying to utilize that framework to try to take care of folks who have these side effects at home. And that's a different model, right? Just like, for example, when I get my vaccine shot, my side effect in terms of pain or fever, it doesn't happen while I'm at the doctor, right? Or it doesn't happen while I'm at the CVS or the pharmacy. It happens when I get home, right? And so you can imagine with an immunotherapy, if I'm having a more potent immunotherapy, now I'm stuck at home with this side effect and I don't know what to do. And I'm going to talk about what we're trying to do to help folks in addition to the research you just heard about. So there are many logistical barriers to clinical trials, and including clinical trials for immunotherapy. Part of this is we didn't know all the side effects for the immunotherapy studies, so we would go through all of them. And really, I always like to folks, when you participate in a clinical trial, you're going to get at least the standard of care, right? The institutional review boards do not allow us to give below the standard of care. And that's the right call, by the way, having sit on some of these boards. And then secondary, you actually add more people to your care team, right? So now the clinical research coordinator is involved in your care. There's actually a company looking at, or a sponsor looking at all the data across all the sites. So you've actually added folks to the care train, so to speak. But fundamentally, the most important thing is to offer folks, not for us as docs to gatekeep, but to give folks the option And this is actually a particularly poignant point for pediatric clinical trials, right? So we do clinical trials for kids as well with immunotherapy over at Rady's. And so making sure that both parents are involved in the decision as well as the child, who may or may not, depending on the age, have understanding of even their disease, let alone what the clinical trial is. Now, the area I want to talk about from here on out, now that we know the problem with clinical trials more broadly and how we're trying to address it and make it safe for patients, is what we're trying to do with immunotherapy, because it requires us new approaches in terms of how we think about the immune system. As mentioned, we had diseases at a 0% survival rate at 5 years. Metastatic melanoma, outside of IL-2 responses, lung cancer. 5 years if you had stage 4 disease, 0.5% survival, right? Now, for melanoma, it's about 40%. In lung cancer, it's about 25%. Triple negative breast cancer went from 0 to about 10 to 15%. So we're moving the needle here slowly, not as far as we like. We want to get to 90, 95, 100%, but it's a lot better than where we were. And so we're going to go to 5 years. I have patients in my clinic from clinical trials from when I first started on faculty a decade ago, right? And they're still doing great. And so that's what we're trying to do. I had a patient I saw last week. He was in his 20s when he got diagnosed. And we actually had to do the first fertility for his donation because he wanted to have kids and he was in remission. And we had to figure it out, right? We had to wash the sperm, and we had to just do everything. And now he has twins, right? And I just saw their Halloween photos and it's great, right? And so you have to learn while you do in this field, right? Whether you're learning in the basic science lab or you're learning in the clinical lab. And so what we're trying to do is raise that bar from 20, 25% to 90, 95%, 100%. What that entails is us understanding each person's tumor. And this is what's precision medicine. And when we look at precision medicine approaches, someone's cancer can either have complete immunologic ignorance. That means your immune system, it's blinded to the cancer being there. So how do we wake up your own immune system, or do we have to engineer an immune system? You may have heard of CAR T cells, right? This is cellular engineering. This is what we're doing if your immune system won't do it, we're going to give you an immune system that does it. This group has immune cells there, but there's kind of like a moat blocking your own immune cells from attacking the cancer. We've actually solved this problem for the most part. This is immune checkpoint blockade. This is where anti-PD-1 approaches. You heard about nivolumab, pembrolizumab for triple negative breast cancer. We can basically bypass the moat. We put a drawbridge over the moat with these drugs that you've heard about and probably seen way too many ads for while watching football or golf. Sorry, what am I saying? And then you have immune tolerance, right? So you have immune cells within the microenvironment that kind of are asleep to the cancer. They're kind of dozing off. They're like the patrolman that's dozed off. How do you wake these guys up? And then you have kind of the rabid dog immune system. You have intrinsic induction. The immune system is just active, but it's not attacking the cancer. It's doing all the bad things you heard about in the prior lecture. And so how do we understand a patient's genetic profile and immune profile to direct the immune system to do something useful rather than something harmful? One way is cell-based therapies. We actually make these locally. The California Institute of Regenerative Medicine, which it was a bond measure that was passed years ago, funds us to do this kind of work as well. So this is your taxpayer dollars at work, so to speak. And it's around cellular engineering. So those folks who aren't in that lucky 20, 25% that their own immune system recognizes the cancer, now we can actually take out their immune cells, or now there's something called in vivo CAR, where you actually give them a drug that actually re-engineers their cells in their body, not even outside the body, the way CAR T works. And you can engineer all sorts of immune cells. You can engineer dendritic cells, you can engineer T cells, natural killer cells, macrophages. So we are actually re-engineering immune systems to fight their cancer now. And actually, this is spread to other diseases. So the first non-oncology use of these drugs is actually going to be for rheumatologic disease, autoimmune disease as well. And it's basically the exact same drug that's used for leukemia and lymphoma. And so this isn't just an oncology story. This is a story of medicine, and hopefully it'll help more and more people. We actually have clinical trials in multiple sclerosis for the exact same type of modality, as well as Crohn's disease, inflammatory bowel disease. And so our goal is really to find these next generation therapies, which we can only do in clinical trials, but to do it in a way that's safe for patients. And this is an area I wanted to focus on clinically. This is an active clinical trial that I have. And so you heard about all these autoimmune side effects that can happen, but they happen at home, right? They don't happen in the infusion center. They don't happen while the patient's in my office. They happen in a patient's home. And so I tell this story when I first got COVID during the pandemic. and I was kicked out of the bedroom and went to the guest bedroom for about 10 days, I think, until I cleared. It got kind of lonely, right? You're in the guest bedroom, you're with your laptop, you're kind of in isolation, or I put myself in isolation because I didn't want to expose the rest of my family. But what was interesting was 2 days before I got sick, my Apple Watch just kept beeping at me, and it kept saying your heart rate's fast, and I'm like, it's broken. I actually had an appointment to go to the UTC Apple store to fix my watch because I thought it was broken. And I was supposed to go that day, and then I just had a fever. And I'm like, huh. And then I did the COVID test, and I had COVID. And so as I sat there in the guest bedroom, I thought, did the Apple Watch predict I had COVID before I had symptoms? Because I'd obviously canceled the appointment. And it turned out it probably did. So I sat there, alone in the guest bedroom and wrote a grant. Because what else are you going to do when you're isolated in the guest bedroom away from all your family and not able to go to work? So I wrote a protocol using wearables, and we actually expanded it, right? I didn't want it to just be Apple Watch. You can use Google Watch, Fitbit, the Oura Ring, if any of you guys have seen or heard of it. And the goal really is to take what helped me, if I'd known more, around wearables, but to use this towards cancer immunotherapy toxicities, because those, remember I showed you earlier that immunologic ignorance, we have clinical trials of new drugs that try to inflame the tumor, right? In the same way a vaccine can inflame you, these therapies cause different kinds of side effects called cytokine release syndrome. You get a neurologic syndrome called ICANS. But broadly, if you're inflaming the tumor, you're inflaming the person as a side effect, right? That's the friendly fire. And so can we monitor this? Can we intercept this before it becomes a problem? Because we have nice drugs that kind of tamper that inflammation. We're the first to integrate this information to the electronic medical records. So if you sign up for the protocol, Apple funded my protocol. They're very kind, and they actually provided us watches as well as some infrastructure support. So we actually have it in the EMR. So now if you say, hey, something's wrong, I can go in the EMR and actually see from every 15 minutes what your heart rate was doing, what your pulse ox was doing. So this is the first step to bringing care at home, because otherwise we're just talking on the phone and you're telling me you feel sick, but I don't have any data, right, to make a decision. Here, at least I have a little bit of data, right? And then it's an ongoing study. We actually have I was in a meeting, we have a prospective clinical trial to use this, where we not only get the information pulled remotely, but we actually have a nurse call based on certain biometric signatures, right? So you don't want to call all the time if you just took the watch off to take a nap, right? But you do want to call if you fell, right, and you lost consciousness, right? And so how do we, or at least your caregiver, get that call? So we're actually working on that clinical trial to see if we can actually prevent folks from having to be in the ER, going in the hospital, and we catch these side effects earlier, right? Just like I potentially, if I had intervened maybe with Paxlovid or something earlier in my COVID, maybe I had a shorter course, who knows, right? So with oncology, this is something we're testing. And we think also, I mentioned Imperial County, right? And Riverside County. If you live 2.5 hours away from where your doctor is, you can envision having the ability to have this kind of monitoring can really impact positively your ability to feel safe while you're on a clinical trial or on this kind of treatment. So I'm very keen on this approach, and especially as we're developing new types of immunotherapy, right? So immune checkpoint blockade, the drugs we talked about, those have a certain cadence of side effect or are pretty good at managing them. But these brand new drugs that are going to take the bar from 25% to 90%, these drugs have unknown side effects, and having that ability to monitor those patients, especially in rural settings, I think may have some benefit. And so we have a prospective study here. And so it's as much about the biomarkers as well as the biometrics, right? You've heard about both today. And so, from these clinical trials, right? How do you find about clinical trials at all? You can ask your doc. There are tumor boards where we talk about these all the time. It's an email address for my disease team clinical trials, including the one that I mentioned with the biometrics. And happy to, I think now we can take questions for both of us, right, Sonia?
Yeah.
Okay, perfect, thanks.
So thank you both for your time today and for 2 outstanding talks and serving for us featured presenters. And at this point, I'm just going to open the floor to you, to the audience, for questions.
No pressure.
I
Oh,
guess
sorry. Yeah.
My logic.
Yeah, exactly.
I have a question for Dr. Patel. Thank you for the very informative presentation. My question is there any mechanism where patients with lower grades of tumors can be assigned to a new immunotherapy clinical trial? Because most of the time what happens is that when they get most of the standard care and established immunotherapies and they don't respond, so the tumor is already in high grade, they go and try new trials. Most of immunotherapies don't work at that stage, but if they get these new immunotherapies in lower grade, they might work. So is there any solution for that?
Yeah, it's a good question. So basically, can we use immunotherapies in earlier stage disease to have better outcomes? And this is an area, so we call this adjuvant, meaning after surgery, or neoadjuvant, before surgery treatment. I think actually your work is with triple negative and I-SPY, if I think I saw this slide, which is a neoadjuvant study. And then we have similar studies in lung cancer, MSI colorectal cancer, melanoma. And so the idea here is when your lymph nodes are still there, right? You haven't cut out the lymph nodes, you haven't radiated the lymph nodes. The lymph nodes are kind of like the barracks for your immune system, right? And so historically, we've cut out the barracks along with the tumor. That's kind of like dropping a missile on your own troops while you're trying to take out the bad guy, right? And so so now we're getting a little better about this, and we're giving the immunotherapy up front, so the immune cells can actually act while there's a lymph node to kind of station the garrison at. And then we actually do the surgery. And in fact, the radiation plans for those patients who don't get surgery, now often they're trying to spare the lymph nodes so the immune system can act on it. So absolutely the goal is to try to use these earlier because you actually enhance cure rates. You're actually curing patients with immunotherapy in stage 1 through 3 disease. In stage 4, there are some patients who are cured, meaning 5-10 years of remission, but it's not as high a rate. And so I absolutely agree we want to use these earlier. But when we're trying to cure folks with immunotherapy, the risk-benefit calculus is a little different, right? If you get a very severe side effect, you always wonder, well, if I intervened sooner, would it work better? Did I need to give immunotherapy at all? And so these kinds of monitoring approaches or having a good biomarker like LC18.2 or LP18.2 is a really good thought because we actually don't have any blood tests to determine if someone has an autoimmune side effect, if they're protected from, or even if they have it. And so we call clinically a diagnosis of exclusion. So to have a blood test that either shows someone's protected or higher risk may be a way of risk adapting, right? Who needs monitoring, who needs more wearables, who needs more patient visits, things like that. But yeah, absolutely. Early stage is where we're curing folks with these tumor types, and we're trying to use these drugs more in that setting.
Hi. You had mentioned in the very beginning that LPCs, is it LPCs?
Yep.
Lower inflammation response?
Lower neutrophil. Oh, that they're anti-inflammatory?
Yes.
So that was something special that we saw in our study, actually. We saw that LPC levels were inversely associated with neutrophils, but not other immune cells. So we think that these LPC species are not broadly anti-inflammatory. They're specifically targeting neutrophils, which means that our immune checkpoint blockade therapy, which activates T cells, that effect should be unaffected.
So how can someone, with that in mind, how can someone increase their LPCs in their body? Because we all walk around with some low chronic levels of inflammation in our bodies, I think. And if this is something, I mean, I'm just trying to, one seems to help the other.
Mm-hmm.
You said it comes from fat that we ingest, or is it fat that our body creates? Is it
Great question.
cholesterol?
So it comes from both sources. We produce it in our liver, and we can also obtain it from dietary sources. So in our mice, we're trying a supplementation just with LPC 18.2 that we've bought, but we're interested potentially with our mice as well as trying an LPC-rich diet and seeing if that can help prevent immune-related adverse events.
Can you give me an example of an LPC-rich diet?
Yes. Sorry. Walnuts.
Walnuts. So it's hard healthy fats.
Yeah, they're part of like what we did a very limited diet study to make associations using a study where the study designers had basically had each patient write down a log for the last 24 hours of what they ate. and they put those into classes like fats, vegetables, gross. And we did measure these patients, their LPC levels, and see did they change in association with specific foods. And as you might predict, it changed with fats. And so, cookie, cheeses, those all increased globally LPC because they're complex fats. But we found that certain healthy fats were more associated with LPC, like nuts, walnuts, certain types of fish. And we looked at, for example, folks who are on a type 1 diabetes diet, which is essentially low fat. And I think this is important. That's a globally low fat diet. They had lower levels. And this might tell us something about these diets, how they might have to be tweaked to be lower, perhaps in saturated or unhealthy fats, while retaining those unsaturated, healthier fats like healthy.
And I think I heard cookies, so folks can get them outside. And I think, but this actually is a big point for our cancer patients. Probably one of the two most common questions they get asked is, what do I eat, right? And if you're a patient, you're not hungry in the first place. So whatever you suggest, you're spending a limited number of effort on trying to eat something. And so when you think about cachexia, which we call the kind of weight loss we get with cancer, thinking about how we can steer nutrition in meaningful ways is really underrated, because when you're not feeling well, when I don't feel well, I don't feel like eating, right? I just want to sit there and rest. And so when we get a possibility of something nutritionally we could do with medicine as food, it's always an opportunity. Sorry, I think. Yeah. Oh, sorry. Oh, just go ahead, and then go ahead, and then okay.
If you have a patient that's having an autoimmune response, are you doing infusions of this LPC IV, or how are you treating them?
So we're not there yet. In our mouse model, we're administering LPC. So we buy LPC that's been purified, and we inject it systematically.
So far in the preclinical models, it's been injectable, and Alicia's actually testing a new topical formulation in a cream, basically.
Yeah.
So that it would just be like a cream formulation that allows, because it is a fat. So, and creams can solubilize with with fat basically, and so we're using that to see if we can get a more efficient and less invasive way to supplement.
First, thank you, Dr. Patel, Alicia, for your time, to take the time to talk to us and present this informative, very important presentation to us. I know you have busy schedules, so
Thank you.
I have so many questions. I can keep you here probably for the next, week. Thank you, Alicia, for your interest and amazing progress. And actually, my question is for Dr. Patel today. Why stage 4 cancer does not respond well to these immunotherapies?
Yeah, it's a great question. So stage 4 cancer typically has a lower response rate to immunotherapy. And folks think this may have to do with what's called immunoediting. So what happens is if a tumor is there for a long period of time, you kind of get this homeostasis. So the cancer is selecting for an immune system that allows it to grow. So it's a bit of a selection bias. And those immune cells that otherwise would have controlled the cancer, they kind of get deleted over time. The other thing is once cancer starts to grow in stage 4, it's more heterogeneous. So it's not just a single target, it's 3, 4, 5 targets. You're asking an awful lot from a single immunologic arrow to hit these different tumor clones that are all slightly different. And the third is probably more metabolic or immunometabolic, where as you have stage 4 cancer, and there's certain organs where you get more immunosuppression, for example, with liver metastasis, you start to get hormones secreted, the same ones that cause you not to want to eat, also cause a type of inflammation that make your immune system not to go well. So it's really all of the above. And so thinking about ways to reverse that inflammation, but also immune strategies that make cold tumors hot, like I was showing in that slide with the 4 aspects, are ways we can try to at least make the prognosis more like earlier stage disease.
So the monitoring you mentioned is very important. I agree with you. This is probably going to be the future, I think, for any diagnostics, medical diagnostics, and monitoring cancer patients. and their treatments and the response to their treatments. Do you have established already any type of devices in addition to the ones that are available on the market today specifically designed? Are you working with a group of doctors and tech companies to design and improve this kind of monitoring for you? And what are you looking for besides the heartbeat? What kind of parameters are you monitoring?
Yeah, it's a great question. So the fundamental debate, and you alluded to this nicely, is do you try to create a device that does it all, or do you try to use the device that people kind of already have? And we actually looked into both. Deploying a new device has a different threshold, and then there are different versions of that device. And then just like you may hear, like Apple AirPods now do, different cardiovascular readings. Even the EKG was a newer reading. The pulse ox, the O2 sat was turned off and on based on a patent lawsuit, for example. These things are dynamic. And so our strategy for my study and for UCSD has been to try to pick biometric parameters that the majority of the wearable devices capture, because the device that is most likely to be worn is the one you're already wearing, was the logic, right? So if we try to create a new device, the fear was, well, someone already loves their Apple Watch or their Oura Ring, now making them wear a second ring or a second watch, and who wears two watches kind of thing, right? And so our logic was more trying to take data from what people are already wearing, because some data is better than no data if they're not going to wear it. But to your point, we've been trying to get 24-hour coverage of biometrics. So if you have an Oura Ring, that thing lasts basically a week, right? But if you have an Apple Watch or a Samsung Watch, there are two types of wearers. They're the daytime wearers and the nighttime wearers, right? And you get different signals, right? The daytime wearers, you get a lot of exertional biometric data, how many steps you can do, heart rate variability, VO2 max. If any of you guys keep track, these are good biometric parameters. But you don't get any sleep data, right? Because you're charging your watch at night. Meanwhile, the folks you get really good sleep data on because they're wearing it at night, you don't know how many steps they took during the day, right? And so what we're trying to do is complete that picture by using multiple devices. But the idea there has been ring plus watch. Or we actually are working with one of the UCSD professors. They have an insert for the pillow, and the pillow detects the quality of your sleep, because the pillow, you can charge it while you're waiting. So we're working on ways of trying to get 24-hour coverage. But I think to your point, you asked an excellent question. We were debating this years ago when we were starting, and the goal was, rather than give a device that people may variably use, to try to create a system using AI informatics to take whatever device they're already wearing to kind of smooth out the data and be able to help folks with the device they're already wearing.
Can you use cell phones, for example, and kind of wireless transmission, I guess?
Absolutely. In fact, all the devices, so a lot of the biometric data you get from the Apple Watch or from, let's say, your Google Pixel or your Samsung is actually your watch plus your device, because there's some biometrics you actually need both for. And so it's absolutely, and that's actually how most devices communicate. I mentioned to the EMR, it's actually generally the phone is plugged in, because there's no interface on the watch, especially the ring. Yeah.
One last question, and I promise I'll let you be to all doctors in the room here today. Are you aware of any cancer vaccine research advancements to help us prevent the disease, not just treat it?
Yeah, it's a great question. There are a couple mRNA-based vaccine studies, which, if folks read the news, have been reimagined with different terminology. They're called personalized cancer treatments now, as opposed to cancer vaccines, in terms of the regulatory path and grant funding path. But broadly, these are hot areas. And in terms of prevention, they're looking in particular, there's something called Lynch syndrome, which is young folks with colorectal cancer, typically, and some other GI and gynecologic tumors. They're looking at trying to prevent there. And also there's some clinical trials looking at germline BRCA, right? So that's the breast and ovarian germline syndrome. But those trials are very early, and it's going to take a long time to figure out. Yeah.
Do you believe they're well funded in that industry?
They were up till about 18 months ago. And then anything with the word vaccine has had a struggle to get funding. And so folks are trying to adapt. Yeah.
Thank you so much.
Yeah. But prevention is the best form of treatment. I couldn't agree more. And vaccines against cancer are one of the treatments along with imaging, right? Like mammography, AI-assisted mammography, AI-assisted CAT scans, AI-assisted MRI, full body MRI, which you can now walk into a mall and get.
Just where this kind of monitoring to prevent also can work and be used.
Oh, absolutely.
Early detection.
Oh yeah, early detection and prevention. If you look at if you can only do 5 things to decrease cancer, right? It's basically no one smokes, you don't drink in excess, you manage obesity, you do optimal screening, right? Mammography, colon cancer, colonoscopies, whatever you need. And then the 5th actually is the most interesting. It turns out folks who have strong social circles, and you guys are obviously a very strong social circle, do much better than those who are isolated. And actually in societies in which either multiple generation families live together or they know their neighbors really well, they always do better from a cancer prevention standpoint. Because, and the question always is, are you just a healthier lifestyle, is your mentality, or you just have someone who can just take care of you earlier on rather than finding someone too late? And so that's an open question, but those are like the 5 things you can control.
We're going to take one
And
last question.
eat cookies, apparently. That's number 6 from Sonia. They're all outside.
Yes, Dr. Patel, thank you for your presentation. In someone that has stage 4 lung cancer, are there different immunotherapies that they could use?
Yes, and so with stage 4 lung cancer, and in particular metastatic non-small cell lung cancer is the most common tumor type, the most important thing we do is we profile each patient individually. And those patients that, let's say, have an EGFR mutation or ALK rearrangement, their best treatment is a targeted therapy. It's actually not immunotherapy. And I have patients on targeted therapy, pill alone, who are on it 8-9 years later, right? If you match the patient to the right treatment. So even targeted therapies in the right setting work really well. That said, the most common treatment in the metastatic setting for non-small cell lung cancer as those long-term type responses is immunotherapy. These typically are what we call PD-1 or PD-L1 inhibitors. You may see the ads, Keytruda, Opdivo, a bunch of these. And then there's some other drugs that target CTLA-4, for example, we sometimes use in a particular setting. And so we know which way to go based on the patient's tumor profile. We look at their PD-L1 score, we look at their tumor genetics, and we also look at the patient, right? Our treatment recommendation for someone who is, 45 fighting lung cancer is going to be different than someone who's 95 fighting lung cancer in terms of what we can safely give as well. This going back to the whole risk-benefit calculus.
I'm going to cheat and I'm going to ask one more question really quick. It's a yes or no probably.
Okay.
And I wouldn't have asked this except talking to Helen.
Yeah.
She mentioned that before entering clinical trials, it used to be that you had to do chemotherapy first.
Yeah.
And she said now that's not always the case.
Yeah, you're absolutely right. So historically, when you're looking at a clinical trial, it's about have you at least tried the standard of care? And back then, the standard of care was chemotherapy. But now we've raised the bar, and the standard of care often is a targeted therapy or immunotherapy. And so those rules have changed over time as the therapies have evolved. And then the logic there is the IRBs and the FDA is worried about folks who could have benefited from one of those therapies kind of getting skipped over and going straight to a clinical trial, as opposed to having at least exhausted one line of standard therapy. So it used to be that standard therapy was chemo, but now it's typically immunotherapy or targeted therapy. Yeah, that's a great question. Okay. Thanks.
Let's take Keytruda.
Sure.
It's pretty widely used.
Yeah.
Discovery and commercialization.
Got it. So with Keytruda or pembrolizumab, it actually has an interesting story. So Merck, when they acquired Schering Plough back in the day, they got a set of drugs, which included a drug called lambrolizumab. It was on the shelf. It was actually literally on the shelf. So what happened is BMS with Ono Pharmaceuticals, which is a Japanese company, had very promising data for what the drug that became nivolumab. And basically, Merck found it on their Schering Plough kind of box in the garage. And they renamed it pembrolizumab. They developed it. They had a large study called Keynote-001, which was the largest phase 1 clinical trial ever conducted because it went across multiple disease types. And they found that it worked in a variety of different tumor types. And so it is, I still believe it's the number one oncology drug, like in terms of market share and things like that. But it's part of a class of therapy called immune checkpoint blockade. But there, sometimes being the first follower works better than being first, right? And that's a classic example there.
Well, I'd like to thank you again for being part of this special presentation today, the first of its kind and hopefully the first of many more to come, bridging research in medicine, translational advances in cancer immunotherapy. Again, we're really grateful for your time, your curiosity, your partnership with us through the Heights Golf Club. And progress in science, again, you can't stress this enough, doesn't happen in isolation. It happens when community is engaged in such as this. It really does take a community, including scientists, including engaged, informed folks as yourself, the patients, their families, our clinician, our advocates, our funders, community stakeholders who are all working together for a life without disease. And so as we head into the atrium for lunch, I encourage you to please continue these conversations. This was a great, lively, engaged question and answer session, and just to connect with us. So thank you all.